Okay, so ladies and gentlemen, you are welcome. So actually, I think if you look at the time is really going, and we have covered a lot in this biochemistry. So that is why I feel like it is actually now the right time to start revisiting some of what we have actually covered in the course. And I hope this video will really be important to all of you. Okay, I Toshi Fish, you are welcome. So, please, I want one of you to actually send a question paper, the past question paper in the group, so that at least we can quickly look into the question paper. Please send the past question paper in the group so that we can actually go through it and solve some of the questions. So that at least I think it will be very important to you for you to know how to approach some of the question. Please send the question paper. Okay, Fallen Akin Willow, you are welcome. Promise Race, you are welcome. Please send the first question paper on this course to the group so that at least you can quickly go through it. Send Biochem 241 past question paper in the group so that we can easily go through them. Please do that now. Send the past question paper of the course now. I'm waiting for you to send the first question paper in the group. Or are you telling me you don't have first question paper, please? Send past question paper in the group so that we can go through the first queue. Okay, good evening. How are you? You are welcome. Blazing, Benter, Joseph Messi, you are all welcome. Aisha Abakar Ibrahim, good evening. Please send the first question paper in the group, in the WhatsApp group. I'm waiting for you. I want to go through the first question paper. I think it would be the best to revise the course. I'm waiting for you to send the first question paper. You don't have first question paper. I'm waiting for you to send the first queue in the group. Send past queue. Okay, so since you don't have the first question paper, so let's just revisit it like this. But I wish that you have the first question paper so that we can go through it and see how you can answer the question. Okay, so thank you. Hajja Aisha just sent one of the first question papers. So let me open it and then we can go through it. Excellent. So let's actually start. This is the first queue. So we are going to so yes, Aisha, I just saw one. So let me go through it. So I think okay, but we can 241. Okay, so good evening. So I have one of the question paper. The question says highlight the relevance of biochemistry to drug discovery using a specific example. So I think You can all see the question. Yes, sir, we don't have first question paper. Yeah, but I think I just have one here. So I think I'm going to solve this one. No, I already have the first question paper. So I think let's just go through this without wasting much of our time. So I think briefly, maybe you can have glitch in network because I'm going to use a bot to solve the past question paper. I think that would be the best. So let me use bot to go through. Okay, so please welcome back. And I think now you can tell them that in the group that the network is now stable. So I actually just want to use both to answer some of the questions. So please, you can tell them that the network is stable now. So let me actually share the question again so that we can read the question together and then we actually solve the question together. So I think let's start with first. If you look at the question paper, you have, and the question paper you have about, let me actually count the number of questions. We have question 1 to question 6. So we have question 1 to question 6, and we are to answer question 4 from the instruction. So you have about 6 questions, and you are to answer the 4 questions out of 6. So I think you can read the question very clear, that the question says highlight the relevance of biochemistry to drug discovery using a specific example. So I think you read the question. The question says highlight the relevance of biochemistry to drug discovery using a specific example. So I think, ladies and gentlemen, there are things that we need to understand here very clear. Number one, we are talking about, this is biochem BCHM 241, and the question is actually the relevance of biochemistry in drug discovery. So although this is actually some of the things that we have not actually discussed about, but I'm actually going to talk about it now. First, in drug discovery, we need to understand, because we have started proteins, but we didn't actually enter into the enzyme. But we should understand that in this, at this point, we are going to look at some concept of enzymes. Enzymes. So what are enzymes? That is the important things. First, enzymes are proteins. In fact, 99.99% of the enzymes, they are proteins. 99.99% of the enzymes, they are proteins. So I think, so what enzymes do? They catalyze the rate of the reaction. They catalyze the rate of the reaction. When we said they catalyze the rate of the reaction, it means that enzymes convert a substrate to product. And very importantly, there are reactions that they can take a millions of years. Let me just give example. So enzymes generally catalyze a reaction. So in a reactions usually that take place in the body, You have a reactant, let's say you have A plus B, and this A plus B will be converted into C plus D. So what converts this A to B is what we call an enzyme. So enzymes, they are actually biological molecules, usually proteinous in nature, that catalyze the rate of the reaction. And more importantly, ladies and gentlemen, when we are thinking about enzymes, there are reactions that they can take millions of years. There are reactions that can take days. There are reactions that can take months. But because as a result of this reaction, or because at the presence of these enzymes, the enzymes can easily convert the substrate into product. So generally in a reaction, we have what we call a substrate, the substrate of the reaction, and then we have products. So whenever you see a reaction, like for example here we have A plus B, so it means that A and B here are the substrate of this reaction, while C and D is the product of the reaction. So what are the substrate? The substrate are the starting material for a reaction. And what you have at the end after the reaction is what we call the product. And how this reaction takes place in the body. Generally, that is what we call a reaction profile diagram. Reaction profile diagram, look at it very clear. In some of my videos, I actually explain that. So in this reaction profile diagram, we have reactant. So our reactant usually come here. We have A plus B here, and then we have C plus D. I just actually want to describe how biochemistry or the relevance of biochemistry in drug discovery, how you can use the knowledge of biochemistry to develop drugs, how you can use the knowledge of biochemistry to come up with either antibiotics or any drugs. So that is why you need to understand the concept of enzymes briefly. And it's part of what we are going to discuss in this course. But I'm going to summarize it here before we actually have the full lecture on the enzymology. But here we are trying to solve the past question paper, and I will show you, or I will actually help you on how to approach some of these questions. So as I'm saying, please go and tell those people in the group, let them be present in this lecture. I realize that it's only 21 of you. Please go and tell them in the group that they should join this lecture. Tell them that they should join this lecture. I have only 21 here. So go and tell them, please. Okay. So what I'm saying is that this is what we call the reactants, and this is the products. And this is what we call from here to here, this is what we call activation energy. This is what we call activation energy. I just actually want to show you how this reaction works, or how enzymes work in actually facilitating the rate of the reaction. So if you have A plus B, before this A plus B converted to produce C and D, the A and B must collide. They must undergo what we call collusion. And the collusion between A and B will lead to the generation of energy. And that energy must be sufficient to cross the activation energy. So therefore, before the reaction takes place, the activation energy must be crossed by the reacting particles. So therefore, they must provide a sufficient energy. So how these enzymes work? So this activation energy is like an energy barrier. It's an energy barrier that must be overcome before reaction takes place. So here what I'm saying, ladies and gentlemen, we have A and B trying to convert to produce C and D. And how this A and B convert to produce C and D, they must overcome this activation energy. And what is this activation energy? Is the energy barrier that must be overcome before you have the substrate converted into product. So what this enzyme does is that it lowers the activation energy. So instead of the reacting particles to struggle instead of the reacting particles to struggle to get the sufficient energy to cross the activation energy, so the enzymes that is there will now actually lower the activation energy. It will what? Lower the activation energy so that this A and B can easily be converted into C and D. So that is what we call activation energy. It's the energy barrier that must be overcome before reaction takes place in the body. So therefore, A and B, before they produce C and D, they must collide. They must collide head to head, and the collision must be perfectly elastic. And these reacting particles must provide sufficient energy before they cross the activation energy. So instead of these reactive particles to collide head to head to provide sufficient energy for them to cross the activation energy, so on their own, the enzymes will lower the activation energy for them. So you see this one here, we're talking about how you have the reaction profile diagram. So in the absence of enzymes. So let's see if we now add enzymes here, what will happen? So in the presence of enzymes, you'll have something like this. Well, let me just, okay. So in the absence of enzyme, this is the activation energy where A plus B will be converted into C plus T. But in the in the presence of enzymes, this activation energy will be lower. You see, this is the activation energy. So in the absence of enzymes, you have this huge energy or this huge activation energy. But in the absence, sorry, in the presence of the enzyme, look at how the activation energy reduce. So it means that this A and B can easily cross this activation energy with just small amount of energy for them to be converted into C and D. So ladies and gentlemen, you may see a question in this course that how enzyme facilitate the rate of the reaction. So the answer is simple. Enzyme facilitates the rate of the reaction by lowering activation energy. Please get it. How enzymes catalyze or facilitate the rate of the reaction in living system is by lowering activation energy. So ladies and gentlemen, under this enzymology, we also need to understand two important terms. One, we have what we call activators. We have what we call activators. And then number two, we have what we call inhibitors. We have what we call inhibitors. So what are the difference between activators and inhibitors? Activators, ladies and gentlemen, get it? They are molecules that increases the activity of enzymes or that activate the enzymes. That is what you call activators. Please get it, ladies and gentlemen. Activators, they are molecules that facilitate the activity of an enzyme. While inhibitors, they are molecules that block or inhibit or stop the activity of enzymes. So these are the difference between activators and inhibitors. Activators increase the activity of the enzymes, while inhibitors decrease the activity of the enzymes. So I want you to understand, I actually wanted to, because we are starting from this point to understand first what are enzymes, what are activators, and what are inhibitors. And from there, we'll see how you use the knowledge of biochemistry to discover or to come up with a new drugs. So now, you understand that activators, generally when you have activators, as I said, activators generally increase the activity of enzymes. You see, activators increase the function of an enzyme. Then inhibitors decrease the activity of enzymes. Please, ladies and gentlemen, do you understand? Do you understand, one, how enzymes work, the activators and the inhibitors? Please, do you understand? I want to see your response on the comment section. Do you understand enzymes? First, I said enzymes, there are proteins, that facilitate or that increase the activity or that increase the rate of the reaction. There are molecules that increase the rate of the reaction. So if reaction is taking, let's say, 10 days, if the reaction is taking 10 days in the absence of enzymes, if in the absence of enzymes, reaction is taking 10 days, so maybe after you add the enzymes, instead of taking 10 days for the reaction to occur, maybe in 1 day. So it means that enzymes increase the rate of the reaction. Instead for the reaction to take 10 days, it now take 1 day by lowering the activation energy. So the mechanisms of the activity of the enzymes, is by lowering the activation energy. Because for the reacting particles to produce, or for the substrates to produce products, the enzymes, for the substrate to produce a product, the substrates must collide head to head to provide sufficient energy that will make them to jump the activation energy. So that is why it's not in every situation that reaction takes place in the body. Sometimes if these reacting particles cannot provide enough energy to cross this activation energy, so the reaction will not take place. The reaction will not take place if the reacting particles cannot provide sufficient energy. So instead of the reacting particles to struggle, instead of the reacting particles to suffer themselves to produce sufficient energy to lower the activation energy, so by adding the enzymes, the enzymes will help them to lower the activation energy so that the substrates, which is A and B in this situation, can easily cross the activation energy to produce a product. So that is why here, look at it in this reaction profile diagram. You see here, in the absence of activation energy, in the absence of enzymes, you have this huge activation energy. But in the presence of enzymes, look at how the enzymes lower the activation energy. Look at it here. So that means that A and B can easily be converted into C and D. So that is how enzymes work. It works by lowering the activation energy. So someone, you may be asked in the exam that what actually or how enzymes work. So the enzymes work, or the mechanisms of enzymes work, is through the lowering activation energy. Yes, they are activators of the reaction. You can consider it in that way. But they are not activators generally. Rather, they are molecules that facilitate, that increase the rate of the reaction. Instead for the reaction to take longer time, so in the presence of enzymes, they will lower the timing for the reaction. So now, we are now trying to also look at some other important terms of the enzymes, where I said we have activators and we have inhibitors. So what are activators? They are molecules that increase the activity of the enzymes, that help the enzymes to carry out their enzymatic activity. While inhibitors, they are molecules that lower that lower or block the activity of the enzymes. That is what we call activators and inhibitors. And ladies and gentlemen, very interestingly, I want to say this. Most of the drugs, I think 99.9% of the drugs that are available in the pharmaceutical industries, they are either enzymes activators or enzymes inhibitors. And in this class, I'm going to explain that very well, and I hope you will understand. But put this at the back of your mind. Enzymes, they are either activators or, sorry, drugs are either activators or inhibitors of the reaction. So they're either enzymes activators or enzymes inhibitors, most of the drugs that are available in the pharmaceutical industry. But some of you will now say how and why. How drugs can be an enzymes inhibitors. How drugs can be enzymes inhibitors. How drugs can be enzymes activators. That is what I'm going to explain now, ladies and gentlemen. But you have to listen very well. Briefly, I think, let me just use this example to explain how to design drugs. Let's see, headache. Headache is not a disease, rather is actually a sign of the disease. So let me actually explain some important concepts. Arachidonic acid is a biomolecule through in which an enzyme called cyclooxygenase converted to produce what we call prostaglandins. Prostaglandins. Please, ladies and gentlemen, get this, and I want you to understand something very important from here. You see, enzymes, they have what you call active sites. So enzymes are usually something like this. It looks like something like this. So let's say this is your enzymes. So enzymes, they have what you call active sites. The enzymes have what you call active sites. Enzymes have what you call, so this pocket that you see here, we call it active sites. So this is what you call active sites of enzymes. This is active site of enzymes. An active site is a region or part of the enzymes where substrate bind. Look at it. You see here we have a substrate where a substrate bind to it to produce a product. So that is what we call enzymes active site. So what I'm saying, ladies and gentlemen, this is arachidonic acid, and this enzyme is called cyclooxygenase. Is an enzyme that converts this arachidonic acid to prostaglandins. And this prostaglandin, through this cyclooxygenase pathway, produce a molecules that cause headache. The headache that we're talking about that usually produced under stress or under some psychological conditions or under some disease condition is due to the production of these prostaglandins. In one of the videos that we are going to do, part of the lipids chemistry, I'm going to give you a details of how arachidonic acid is used to produce prostaglandins. So prostaglandins, ladies and gentlemen, get it very well. It uses arachidonic acid to produce prostaglandins, and that prostaglandins, there are molecules that cause headache. Prostaglandins, there are molecules that produce headache. Using arachidonic acids, the cyclooxygenase is an enzyme that convert the arachidonic acid to prostaglandins. So now, ladies and gentlemen, assuming now you get a molecules, let's say you get molecules A, because we have what we call enzymes inhibition, and that is what we are also going to talk about when we come to endymology. We have different types of inhibitors. We have what we call competitive inhibitors, and we have non-competitive inhibitors, and we have uncompetitive inhibitors. So that is what I want to, ladies and gentlemen, to know at the back of your mind. Please, all this thing that I'm saying, they are very important because it's part of what we are going to discuss in some of our class. So remember, this is actually a first question paper that we are trying to solve. How biochemistry is used in drug discovery. So first, as I said, arachidonic acid. So with a specific example, so you can give this with the examples. Arachidonic acid is converted into prostaglandin using cyclooxygenase enzymes. And these prostaglandins that produce from arachidonic acid using the cyclooxygenase enzymes causes headache. It causes headache. So now, assuming now you have headache due to production of these prostaglandins. So how can you prevent the production of that prostaglandins in the body to prevent headache? Is simple. You can get a molecules that will bind, especially maybe inhibitors, because at this point you need inhibitors. If you can get a molecules that is inhibitors that combines to the active site of these enzymes to prevent arachidonic acid from binding to its active site, it means that, so if you can get actually inhibitors, here we can set plus inhibitors. So if you have cyclooxygenase, you have arachidonic acid, and you now have your inhibitors. So if you now have these inhibitors, especially in high concentration, when it binds to this cyclooxygenase, it means that arachidonic acid cannot bind to the cyclooxygenase because of the presence of these inhibitors. So it means that if arachidonic acid cannot bind to the active site of cyclooxygenase, it means that there will not be production of prostaglandins, and therefore there will be no headache. There will be no headache. Ladies and gentlemen, do you now understand? That is why I said inhibitors, drugs can either be activators or inhibitors. So you have cyclooxygenase which convert arachidonic acid to prostaglandins. So if you now have an inhibitor, a molecules that can bind to this cyclooxygenase and prevent arachidonic acid from converting into prostaglandins, it means that definitely there will be no headache. There will be no headache. 1 minute. Please, do you understand? Okay, Rosalind said, sir, please repeat. I said cyclooxygenase is an enzyme, it's an example of the enzymes that combat arachidonic acid to produce prostaglandins. And these prostaglandins cause headache. It causes headache. So assuming now you get a molecules that combine to this cyclooxygenase and prevent arachidonic acid from combating and prevent arachidonic acid from binding to the active site of the enzymes to produce prostaglandins. It means that this inhibitor can serve as a drugs for headache. Drugs for headache. So arachidonic acid is converted into prostaglandins. So if you can get molecules that can inhibit this cyclooxygenase from binding with arachidonic acid, it means that there will be no formation of prostaglandins. And if prostaglandins cannot be produced, it means that there will be no headache. So this inhibitor, that is these molecules that prevent the interaction of arachidonic acid with cyclooxygenase, it means that there will be no headache. So this inhibitor can serve as a drug. So during exams, do we need to explain with diagram as you have done here? Yeah, it's the best to do that. But this diagram is just actually to show in the case of how enzymes work. So if someone asks you how enzymes work, you can show this diagram that okay, in the absence of enzymes, you have this activation energy. Then after adding the enzymes, it will now lower the activation energy. And in the case of the reactions, you can now show that okay, arachidonic acid is converted into prostaglandins using cyclooxygenase enzyme. So if you now have inhibitors and this inhibitor bind to the cyclooxygenase, it means that arachidonic acid will not be converted into prostaglandins and therefore there will be no headache. So therefore this inhibitor can serve as a drugs for headache. I hope, ladies and gentlemen, you are now clear and you fully understood the concept. Is that clear? So you see, a drugs or a biochemistry can serve as a foundation. Biochemistry can serve as a foundation for getting the knowledge of enzymology, which can be used in drug discovery. That is the first thing that you are going to start when you are going to answer this question. So therefore, you give an example. Cyclooxygenase is an enzyme that converts arachidonic acid to prostaglandins. And then in the presence of inhibitor, the inhibitor will now bind to the cyclooxygenase, and then arachidonic acid cannot be converted into prostaglandins. Therefore, there will be no headache. So this is actually one of the steps, ladies and gentlemen, that can be employed to answer this kind of questions. So you see, I'm actually taking my time because if you are asked to answer a particular question, especially from my own point, I have to clearly demonstrate on how to answer that question because you need to follow all these steps. Okay, generally enzymes work to lower the activation energy, and some of these enzymes they actually involve in producing molecules that can cause disease. Like for example, arachidonic acid produces prostaglandins, which causes headache. So therefore, getting a molecules that can inhibit or that can block the activity of the enzymes that involve in producing molecules that are associated with disease, then that molecules that can block the activity of the enzymes can serve as a drugs. So it means that the molecules that block the activity of the enzymes are inhibitors, which can also serve as a drugs at the same time. That is why I said most of the drugs are either enzymes activators or enzymes inhibitors. Please, sir, what are inhibitors? Inhibitors are the molecules that block or stop or inhibit or lower the activity of the enzymes. That is inhibitors. So, ladies and gentlemen, I think you now understand how the knowledge of biochemistry can be used in drug discovery. So that is why one of the major role of biochemistry in medical sciences is actually they are involved in drug discovery, in disease diagnosis and disease management. So you can use this. In fact, you can even use this knowledge that I gave you, you can use it to produce a lot of antibiotics. A lot of antibiotics. Let me just give you this example. Some of you, I know that you have done biology, so you have done some basic microbiology from your lower, I think from your colleges of health or colleges of health sciences. So now, let me just give you this example. Remember, bacteria, bacteria generally in humans we don't produce cell wall or we don't have peptidoglycan. So generally, microorganisms uses glucose 6-phosphate uses glucose 6-phosphate, although it is actually a complex mechanism. It's a complex mechanism. It involves so many enzymes. It involves so many steps. So generally, microorganisms use glucose 6-phosphate to produce what we call uh, gly- uh, peptidoglycan. Peptidoglycan. Peptidoglycan using what we call transpeptidase enzyme. Transpeptidase enzyme. I don't need to even write the enzymes because by looking at the word even these ASE, it means that it's an enzyme. So you see, in the process, you know that bacteria they have cell wall, and it's what actually protect them. It is actually what protect the bacteria. So they generally have cell wall, and it's what protect them from any external attack, from any organism, from immune system. Let's say you said that biochemistry serves as a condition in one. It serves as a foundation, not condition, foundation in drug discovery, especially using knowledge of enzymology to actually come up with either enzymes inhibitors or enzymes activators. That is why now I give you examples where you have cyclooxygenase. If you can get the inhibitors of this enzyme, it can serve as the drugs. And also now I'm also trying to give another example because from the questions they said with a specific example. So in this case, you have glucose 6-phosphate, which can be used, although it actually involves a series of steps. I'm just actually giving you some short story because these things you have not done them and we are not going to do them now, maybe in your second semester. So glucose 6-phosphate through a series of reactions can be converted into peptidoglycan. And peptidoglycan is a molecules that are actually found on the bacterial cell wall. And it's actually what protects the bacterial cells. So if you can break down this peptidoglycan of any microorganism, so there is a likelihood that those organisms are going to die. So now, assuming now you get a molecules that can inhibit, the same time maybe inhibitors that can inhibit the conversion of glucose 6-phosphate to peptidoglycan, it means that the bacteria cannot produce cell wall. And if they cannot produce cell wall or peptidoglycan there on their cell walls, it means that they are not going to survive. So that is why, ladies and gentlemen, at this point, the molecules that can use to block the activity of these transcriptase to actually prevent the conversion of glucose 6-phosphate into peptidoglycan, it means that that molecules can serve as an antibiotic. In fact, penicillin and penicillin, most of those antibiotics, they are inhibitors of these transcriptase. They are inhibitors of these transcriptase. So, ladies and gentlemen, I think from here you can see that clearly I gave you a classical examples on how the biochemistry is relevant in drug discovery. Look at the questions. It says that highlight the relevance of biochemistry to drug discovery using a specific example. So I give you two examples here. So you can take any of these two so that you can actually answer a question. So now, highlight how glycoproteins are involved in the ABO blood group system. Highlight how, I think you can all see the questions. Let me increase the size of the question. And I think this is actually part of what we have done from some of our classes. Look at the question. The question says highlight how proteins are involved in the ABO blood group system. So I think I would like you to answer this question. Which of the glycoproteins are found on the surface of the red blood cells? Because you should understand that, ladies and gentlemen, we have different blood group types. We have different blood group types. We have blood group A, we have blood group B, we have blood group AB, and then we have blood group O. So now, what make one blood group differ from another blood group, ladies and gentlemen, is actually the presence of the glycoproteins. So now let me start by asking, which types of glycoproteins are found on the surface of an individual with blood group A, and also the glycoproteins are found on the blood of somebody with blood group B, the AB, and then O. This who can tell me what is the name of the glycoproteins that found on the surface of a red blood cells with blood group A? The name of the sugar. The name of the sugar of someone with blood group A, blood group B, blood group AB, and then blood group O. Please, I need answer to this question. Write it on the comment box. I can see the answer. Please, I'm waiting for you. Answer the question, please. Answer the question, please. What are the sugars that found on the red blood cell? Oma? No, no, it's not fructose, it's not galactose, it's not glucose. I think in some of my videos I mentioned this very clear. What are the sugars that are found? Okay. Yeah, Oma, excellent. You said that A is N-acetyl galactosamine. Excellent. So an individual with blood group A has N-acetyl galactosamine. So if an individual is having blood group A, it means that the person is having N-acetyl galactosamine on the surface of the glycoproteins, and that is why the person is having blood group A. And then, of course, still Omar said, if someone below group B, it means that he has galactose. And then someone with blood group AB, it means that the person has both N-acetyl galactosamine and galactose. So that is what happened. For someone with blood group AB, it means that the person is having both N-acetyl galactosamine and galactose. And then for someone that have AB, it means that both these sugar are present on the surface of the red blood cell. While an individual with blood group O, it means that they have none of either N-acetyl galactosamine or galactose. That is the answer. Is that clear? Yes, it's absent. For O, there is no any of the sugar. For AB, they have both the sugars, N-acetyl galactosamine and galactose. And for O, there is none of the sugar. So this is actually what you are going to use to answer this question. Is that clear, ladies and gentlemen? So now let's move to the next question. I think all these things I have discussed them on my video. We have discussed all this on my videos. So now the next question is saying that what is the basis for using serum aspartate transaminase and alanine transaminase as a markers for liver damage in humans? In humans, you see, some of you will even be surprised in that how where these questions are coming. You see, most of these things are coming from explanation. Okay, someone is writing something. Let me see. Muhammad Rufai Inua, blood group B galactose, blood group O absent. Yeah, we have already done with that. We passed that level. So now question C, 1C. What is the basis for using serum aspartate transaminase and alanine transaminase as a marker for liver damage in humans? So I think in some of my videos I explained this, but I didn't give example like this. No, I think I even give example like this, especially for liver function test and kidney function test. So let me answer the question. So we are talking about S. We are talking about serum that is ALT, and AT. So ladies and gentlemen, to answer this question is very simple. Let's say this is the liver. Let's say this is the liver. Ladies and gentlemen, let's say this is the liver. So these two enzymes, which is ALT, alanine aminotransferase, or AST, aspartate transferase. We can't see, okay. We can't see what you are drawing, sir. Oh, so let me black marker. So let's say this is a liver, ladies and gentlemen. Let's say this is a liver of a particular organism. So in the liver, there are two enzymes, and these two enzymes, they are mainly found in the liver. ALT. The ALT is alanine transferase enzyme, and then we have AST. AST. Or let me just use ST, AT, or AT, aspartate transaminase. These are the enzymes that are present in the liver. They are enzymes that are present in the what? In the liver. These are enzymes that are supposed to be in the liver. Although they have, sometimes you can also find them in blood, but they have their concentration. And I can't remember exactly the concentration of this ALT and AST. But the major idea is to tell you that whenever you find out that the amount of these enzymes in the blood or in the serum exceeds the normal range in the blood, it tells you that there is this liver damage. Because if the liver is really damaged, the content of the liver will release into the blood. And that is why you will see that some content or some amount of the molecules that are supposed to be in the liver, they are going to exceed the normal range. An example is this ALT and AST. These ALT and AST enzymes, they are enzymes that are usually found in the liver. So whenever there is a liver damage, then you expect that the liver is having problem or the liver is damaged. So that is why normally in the basic laboratory or in the routine laboratory, if you want to test whether someone is having a liver problem or not, like for example, let's say, please minimize the question paper so we can see the board clearly. Okay, let me add. Okay. So this is what I'm actually drawing. I said, let's say this is a liver. So in the liver, there are these two enzymes, aspartate transaminase and alanine aminotransferase. There are enzymes that are supposed to be in the liver. So whenever there is a liver damage, when liver is damaged, you remember when we said something is damaged, other mean you have actually a cup and the cup is damaged. So whatever is inside that cup will actually be fall out, or it will actually leave the cup and move into the other environment, or into the environment where the cup is. So the liver is in our body. So when the liver is damaged, the content of the liver will be released into the blood. And some of the enzymes that are found in the liver is this alanine aminotransferase and aspartate transaminase enzyme. So whenever there is a problem with the liver, so there is a likelihood, especially if the liver is damaged, so the content of the liver will release into the blood. So once you go to the hospital and you ask you or you check the ALT and AST in the serum or in the blood, and then you realize that the content of ALT and AST goes very high, so it's an indication that tells you that there is problem with the liver. In fact, this is one of the first steps that's in some basic laboratory that you can start with. Assuming now you have liver problem, or you are in the hospital, or again in the hospital, let's say you are actually a medical lab scientist and you actually want to diagnose someone with the liver problem. So the best thing actually, just get a kit, check the amount of this ALT and AST in the liver. So if you realize that it goes high, it means that there is definitely liver damage, like in the case of hepatitis. But to know exactly what happened to the liver, so you need to go for further tests. You need to go for further tests. So, ladies and gentlemen, this is actually how to approach and how to answer some of this question. And I hope you really enjoy them. And let me look at some other questions so that at least, but I think we can stop here. I think we can stop here. But let me see another things here. So, although there is also D, there is also question D, and I don't think maybe because we already spent 1 hour. So, do we need to continue or we should stop, please? Let me hear from you. And don't forget, ladies and gentlemen, I always tell you, especially those that didn't join the YouTube community, and that is why I said all the videos on biochemistry they are free, but there are biostatistic, there are two physiology. I think I'm taking almost 4 courses, but this course is assigned to me officially. But the other courses, physiology, biostatistics, those ones they are actually videos that if you really want to watch them and you want to have access to them, and you also want to have a solution to the first question papers on those videos, so you need to register with the YouTube membership. So register with the YouTube membership. And tomorrow, ladies and gentlemen, I think we are going to start solving some of the past question paper from I think we have actually started answering. I think we have never answered physiology first question paper. So I think tomorrow I will use tomorrow to start solving some of the first question paper from physiology. So the importance of actually solving some of this first question paper, it will guide you, it will help you, it will assist you on how to approach some of the questions. And there is likelihood in most cases some of the questions that we are going to solve here, you will actually have the repetition of some of them in your exam. So that is why you need to understand them very well. Sir, I tried registering countless times, but I can't see any join icon. Okay, if that is what you want, I will send you the link where you are going to see joint. So you can, I think even here you can check from the YouTube, you will see that joint. So in most cases, actually, if you try your ATM, if you try your ATM and it didn't work, you can use your friends or someone that is close to ATM. You can use his ATM so that you can actually use it to register with the YouTube membership. And I always ask you and I always encourage you to register with that of PQ and short videos. There are videos that are only meant for those people or those that registered at that level. So that is why I want you to register with that of 1,500. And very importantly, as I told you, that if you didn't join the YouTube membership, you are going to miss a lot of videos, and of course, you may not have access to some of our videos that are actually on the issue of sorry, on the biostatistic and the human physiology. So you can use your friends ATM to join. Stop wasting your time because you have less than 1 month to start your examination. So try to join the YouTube membership so that you will not waste your time. I advise you to do that. And ladies and gentlemen, I think I'm going to stop here. Please, is there any question? If there is not any questions, then we are going to end it. So you can use your. And I want to tell you that today there is no class. Just visit this video and then we continue next week. So no question? So thank you very much and see you next time. And I see that a quite number of you have joined the YouTube. Fallen, you are there. Chidiola, you are there. Comfort, you are there. You see, Comfort ma have joined the YouTube. And Lucia also, you have joined the YouTube long ago. You see, even Agnes and Tony, she also joined. So I think you should stop wasting your time and face the reality. And it's because if you don't want to pay the money, then it's your own issue, and I don't have problem with that. I'm not saying that it's compulsory. It's for your own benefits. It's for your own benefits. So it's not compulsory. And of course, for all the videos on biochemistry, you will have problem, and you have access to them all the time. Good evening, sir. Please, we have access to this revision. Yeah, you will have access, especially you that are members. Good evening, sir. I really missed today's because I'm taking emergency right now. No, no problem. The video is still on the YouTube channel, so you can go and watch it anytime, over and over. That is one of the advantages. Sir, can we have access to the revision? Yeah, of course. Let's answer question 1D before you stop, please. Lucia, why? I think to answer question D is also a very simple, because vitamin B, because this also, if you look at this question from beginning to the end, especially question 1, question C, and question D, they are all on enzymology. So she said that she wanted me to answer question D. So remember, ladies and gentlemen, vitamin B, especially we have vitamin B1, which we call it thiamine. Thiamine. We have vitamin B. And we have some of the reactions that require this thiamine. Because, ladies and gentlemen, I forget to tell you something. It's because we didn't discuss about this enzymology. We didn't actually treat the topic. We have what you call coenzyme. Coenzyme. So coenzymes, they are molecules that require by some enzymes. Okay, someone is saying that they have another lecture. No problem, we are still having the classes, so I think we can, or maybe let me answer the questions. You can revisit it again and watch the last part of this. So what I'm saying that when you said coenzymes, they are molecules required by the enzymes for them to carry out the enzyme's activity. So there are molecules that are needed for them to for the enzymes to carry out the reaction. And examples of some of those reactions is when we have like pyruvate. Pyruvate is converted into acetyl-CoA. It's converted into acetyl-CoA using an enzyme called pyruvate dehydrogenase. Pyruvate dehydrogenase enzyme, pyruvate dehydrogenase complex. That is the name of the enzymes. The enzymes, pyruvate dehydrogenase complex, convert pyruvate to acetyl-CoA, and these enzymes require the vitamin B1, thymine. It requires thymine. And ladies and gentlemen, this is not the only enzyme that require thymine. There are a lot of them. There are a lot of them, but I'm just giving these examples. So it means that if there is this deficiency of thymine, some of those reactions cannot take place in the body. Like for example, where we have pyruvate, which will be converted into acetyl-CoA. So do you know the major role of acetyl-CoA in the body? The major role of acetyl-CoA, it will actually a molecules that can undergo what we call citric acid cycle. And the major role of citric acid cycle in the body is to provide a lot of energy, that is ATP. So now assuming this thymine is deficient, or is actually there is a deficiency of this thymine, so pyruvate will not be converted into pyruvate cannot be converted into acetyl-CoA, and therefore energy will not be generated. And that is why the deficiency of thymine cause a disease called beriberi. It causes a disease called beriberi. Why? Because beriberi is associated with low of energy generation. So why there is that low energy generation is because the thymine, which is vitamin B, is deficient, and therefore there will be no enough energy in the body. So you can just give this example. What is the relevance of vitamin B or B vitamins and their derivatives to the catalytic reactions in our body system? So this is actually some of the examples. So remember what we are going to say, ladies and gentlemen, to answer this question. You can say that, okay, vitamin B, there are co-enzymes that require by some of the enzymes in the body to carry out their enzymatic activity. That actually aid, or that are required for the enzymes to carry out their activity. So now, ladies and gentlemen, after you mention that, then you give examples, then you can write examples. Pyruvate is converted into acetyl-CoA using an enzyme pyruvate dehydrogenase complex. The acetyl-CoA produced by these enzymes is very essential for TCA cycle to take place. So therefore, in the absence of this vitamin, especially thymine, the pyruvate cannot be converted into acetyl-CoA, and that will lead to low energy generation of the body, leading to a disease called beriberi. So let me repeat myself. The question says, highlight, let me look at the questions. The question says, what is the relevance of B vitamins and their derivatives in catalytic reaction? So you mentioned that enzymes require co-enzymes for them to carry out their activity. And some of the vitamins B are co-enzymes. So in the absence of, or in the when these vitamins are deficient, so there are reactions that cannot take place in the body. And some of this reaction, you give example. You write the conversion of pyruvate to acetyl-CoA using an enzyme called pyruvate dehydrogenase complex. So this pyruvate dehydrogenase complex require thymine, which is vitamin B1. So in the absence of thymine, there will be no conversion of pyruvate to acetyl-CoA. That lead to a disease called beriberi. You can also give another example where alpha ketoglutarate is converted into into succinyl-CoA using an enzyme called alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase it also require this thymine. So ladies and gentlemen, I think I answered question 1 to D. It took us to 1 hour 14 minutes. Yes, Lucia, I can see that, sir. I was among that first set that joined your YouTube channel. Oh, yes, you are still a member. I can see you have joined. It actually gave an indication that you have joined long ago. Yeah, but of course, look at Obiroma, Mary, Ifeoma. She didn't join. Oma, you didn't join. Chio Maglo, you didn't join. So I don't have problem if you didn't join, especially for biochemistry courses. So I don't have problem. But for the biostatistics, for physiology, if you didn't join, you cannot even have access to the videos. So tomorrow we are going to look at physiology because I already sent you a video on solution to some of the questions in biostatistics. So we are going to do that on physiology, please, tomorrow. Tomorrow. Maybe let's start. I want you to send to me the first question paper in the tutorial group for human physiology 225, please, immediately after this class. Thank you. See you next time.